very good afternoon all. Very happy to meet you all back in this awesome session. So I all know that your DBT exam is very near and definitely you need to practice a lot. Yes, so especially this bioinformatics, it is a very much awesome unit. Yes, because you can very easily and fastly score better marks by using this good chapter. I can tell you that in just 10 days you can complete a very good course on bioinformatics and you can boost your DBT score by much fold if at all you are going to practice these MCQs, right? Okay, yeah, so if at all you are new to me, I am Dr. Sanarnya. I have completed my Ph.D. in Biomaterials. I do have 16 papers to my credit and I have completed my NET and SET and I have coached many aspirants like you for your CSIR examination, DBT, GATE, SET, BET and all. So don't forget to follow me at Unacademy such that you will get the good notification about the courses and classes that I launch for free as well as for the paid version. Right. So without any more delay, let us begin the first question of the day. Right. So question number 1. Which is the default scoring matrix? Which is the default scoring matrix that is being used in BLAST? Okay. Right. Yes, Pallavi. Right. So you maybe if at all you could have studied your bioinformatics in your UG or PG or exclusively you are preparing for DBT. The first point you need to understand is few golden topics. Okay. So among them the one topmost topic which has come much repeated, it is going to be this similarity. Okay. So under the similarity search, okay, so you have two main options. One you have to do with single alignment, another one it is going to be the multiple, yeah Krishna, multiple alignment. Okay, right. So under the single alignment, or you call it as a pairwise alignment, yes, you will be getting two. One is going to be the BLAST and other one is going to be the FASTA. Under multiple sequence alignment, you will get a good example of cluster W, S Jyoti, and then you will be getting another interesting example. So that is what is called as tea coffee. Okay, funny name, right? Yes. So that is for multiple sequence alignment. But under your pairwise, or you call it as one-to-one sequence alignment, you have this BLAST and FASTA. Okay, right. Usually this pairwise alignment, they will follow a local alignment. What do you mean by local? You just compare at your basic level, at a minimum level you used to compare. That is what is called as a local alignment. But when you extend it throughout, then it becomes a global alignment. So, okay, so this is the major difference between local and global. Okay, so now coming back to this question. In this question, they are asking what is the scoring matrix? Scoring matrix means the algorithm. Okay, so what is going to be the scoring matrix that is being used in the BLAST? Number 1, is it going to be the PAM 62? Number 2, it is Blossom 62, Blossom 60, Blossom 80. Okay, so what is going to be all these? These are all algorithms. But among them, which one the BLAST is going to do with? Right. So as I was telling you about this global and local alignment, this if you know the answer you can type or else I'll help out with the answer. Yes. Come on guys. So this is a DBT PYQ. Okay. So this was asked in 2008. Alright. Fine. Okay guys. Fantastic. Come on. Right. So shall I tell you the answer? Alright, never mind. It's a simple subject. Don't panic. Right. So as far as this PAM is considered, very good Krishna. As far as this PAM is considered, it will be only for your global alignment. Okay. Right. But you know this is BLAST. Yes, Shalini and Krishna. BLAST is going to be for the local. What is BLAST? Is going to be the basic local alignment search tool. Correct. So that is what is going to be the blast. So definitely I will eliminate my PAM. PAM is not at all meant for local alignment. It is global fellow. I cannot pick him for blast. Okay, eliminated. Among the given other option, there is no matrix called Blossom 60. Gone. So either 62 or 80. Among the two, which can come? Only the Blossom 62 can come. What do you mean by blossom? Yes, block. Okay, so it is going to be the similarity. So in blocks and blocks, it is going to be similarity. Okay, so that is what is called as the blossom. Okay, right. So this is going to be your block. In block, it is going to get substituted. Okay, right. So all these blossom matrices are only local alignment. So I told you that is how I eliminated the PAM 62 option. Understood? Okay. So if at all it is going to be this Blossom 62. This Blossom 62, it is only obtained from comparing the sequences that have greater than 62% of divergence. Okay, so that is the algorithm that I am going to take up for comparing my blast results. So this Blossom 62, it is what is meant for my blast. Okay, so if I take Blossom 80, what does it mean? It is highly conserved or it is less conserved? Blossom 80 means it is going to be highly conserved. Okay, so 80% it is going to be similar. Got it? So 80% it is going to be identical. Correct? Yes, Preeti. Absolutely. And it is highly conserved. Right. But if at all I am going to take up with Blossom 45 or I am going to take up with the PAM 250. This PAM 250, it is going to be the most divergent fellow. Understood? Yes. So to sum up, yes, we'll just take a few things to just sum up what we have learned so far in this first question. The first question, they asked what is the algorithm. Okay, so what is this algorithm that they are going to use it for this blast. Okay, so the correct answer, it is going to be your Blossom 62. Okay, right. Then we saw what is going to be the two matrices. One is going to be PAM and the other one is going to be this Blossom. Okay, I told you that Blossom is nothing but it is the block substitution. Okay, so if the number is higher, okay, so if the number is going to higher, it says that it is highly conserved. Okay, right. So as the number goes up, it is highly conserved. So which is the maximum conserved fellow? It is going to be the Blossom 80. In the middle, Blossom 62 is there. That is only usually used for all the local alignment. That is going to be your blast. Understood? Right. The next one we saw was PAM. PAM is point accepted mutation. It is going to be a mutation. So how should be a mutation? If at all my thing is highly conserved, mutation should be more or less. I am saying that my sequence is conserved. Yes, Kunal. So how should be the sequence? That's okay. So how should be the sequence? It should be less mutated, isn't it? Less mutated. Only one mutation, it is going to be conserved. If at all I am going to say, I have 250 mutation. So can it be conserved? No, this is going to be divergent. Got the point? So in PAM, 1 is representing convergent, 250 is representing divergent. In Blossom, 60 means it is highly conserved. If it is going to be your Blossom 32, it is less conserved. Understood? Yes? So don't trick between the other terms. Yes, commonly Blossom 45 and 250, it is represented as more divergent, and Blossom 62 is what is used in the blast, and Blossom 60 is highly conserved, also this PAM 1. But remember, Blossom is only for the local alignment. PAM it is for all the global alignment. Yes, everybody in the class. So this first question is done. Yes, shall we make up a move towards the next question? Okay, so say the next question. Everybody, come on. So this is also your DBT. Yes, Krishna. Correct. Conserve means there won't be much mutation. See, if I am talking that my histones are highly conserved. What does it mean? Will it change? No. So there will be very, very less mutation. Correct? That is why your PAM is 1. Okay, your similarity is 80. Good, Praveen. Come on. Yes. Got it, everyone? Alright. Fine, Krishna. Yes, Praveen. So this is also your DBT 2008 question. Which alignment is used to predict whether the two sequences are homologous or not? Think, everybody. So if at all I want to see whether it is homologous or not, should I see the overall similarity, or just I can take a little bit region and check? Think, everyone. Is it so? Think of the definition. Think of the definition, everybody. What is the definition for homologous? I'm not talking about homology completely. It is homologous. Homologous means it is in total. Okay, yes, Mona Lisa. See, the correct answer is going to be, you have to think the total. Total means end to end. I gave you a clue now. Sure, Krishna, I'll tell you. End to end you should check. Then only you can say it is homologous. Yes? Yeah, you absolutely should have a same ancestor. End to end it should match. Isn't it? Yes, Kunal, you got the clue. The correct answer is option B. It is going to be your global alignment. Why it is a global alignment? Because you can call them as homologs. That means they have a common ancestry only if they have more than 30% identical in their overall length. For example, yes, so these organisms I and I is equal. Okay, right, but all the things features are different. Okay, so it is having a ear like this. This fellow is having a ear like this. Okay, right. Yes, so can you call them as homologs? Maybe no. Yes, this is having a face like this. This is having a face like this. So minimum 30% it should be identical. So if I take only this region, so this eyes and this eyes, maybe it shows no. So maybe these two eyes alone I check, maybe it will show like 60% homology. Okay, right. So I should not take tidbits. I should take only the overall sequence. If it is going to be overall, the keyword is end to end. If at all I am going to take end to end, everything, the entire sequence from the top to the bottom, I am going to take the entire sequence. Out of the entire sequence, how much is similarity? If it is greater than 30%, then only I can call them as the homologs. Understood now? Yes? So what's wrong with this local? Local means any restricted amount I can take. Okay, right. So here pairwise and multiple, it doesn't convey much sense. Why? Because this is all whether we are going to take it. Yes, this is all coming under again local only. If I do pairwise, it is blast. Blast is again it is coming under local only. Got the point? Yes, absolutely. Got it, Krishna? Okay. Yes, Shalini. So everybody in the class, did you got it? Why only this global is going to be the correct answer? Because it is the overall similarity. It is not about the tidbits that you create, but absolutely this is meaning the overall similarity. Understood? Okay. See how many questions have come in the 2008 paper. Yes, maybe our paper also we may get more questions. So don't skip this unit, guys. Right. Fine. So in molecular dynamics simulation, yes Preeti, the dependence is on Which of the following? Number 1, only position, only momentum, both position and momentum, either position or momentum. Very good, Kunal. Yes. Yes, Krishna. Good, Praveen. Come on, everybody. Keep it up, keep it up. Yes. So, your MD, molecular dynamics, it depends on, Kunal. Great. So, it depends on both the position as well as momentum. That's why I gave the new questions. Okay, so whenever I am making another session, it should not be a repeat for you, all right? Yes, so that is why I used to take some different questions. Very good, Madhu, such that you will get more prospective practice. Yes, good. So this molecular dynamics, it is a simulation. So it is going to involve your physical movements, okay, and also this momentum, okay. So this is going to be equal to R into P. So R is going to be the position and P is the momenta. Got it? Yes. So molecular simulation, it is going to include both them. Fantastic, everyone. Keep it up. Right. So now coming to the next question. In phylogenetic analysis, okay, maximum likelihood method, it is chosen when the sequences have which of the following? All right. Yes. In phylogenetic, okay, so in phylogenetic analysis, the maximum likelihood method, it is selected whenever the sequences are having. See, I am not talking about the parsimony, I am talking about the likelihood. Name itself suggests. Likelihood. Good hint. Think and answer. Is it? Think. No friends, not at all. See, when I say it is maximum parsimony method, what you will say? If I say that there is a maximum parsimony, then what you will say? Parsimony is only least change, no? Yes or no? Come on. So least change means, yes Praveen, good. So parsimony means only it is the least change. So definitely this is not the answer. Got it? Yes, so I cannot have it for strong similarity, because strong similarity means it is going to represent your maximum parsimony method. Gone. So maximum likelihood. Likelihood means there is no clear identifiable similarity. That is what is called as likelihood. Maybe like that we'll say no. Likelihood of getting the correct one. Yes? Okay, fine. So you have to understand that there are different methods. This is all the methods that are available for construction of your phylogenetic tree. Under that, the first method is called as distance based method. So under that you have two things. One, it is ujima and neighbor joining. Okay. So under your phylogenetic tree. Okay. So phylogenetic tree construction method. The first method is going to be the distance based method. Under the distance based method, remember the first one, it is going to be the Ujima. We could have done few problems on this also. Ujima based method. Correct, no? Yes. Next is going to be NJ method. Nearest neighborhood joining method. So these two are coming under the distance. Likewise, you have the character based method. So under the character based only, you have the maximum parsimony. Maximum parsimony means maximum similarity. Then maximum likelihood. Likelihood means maybe this may be right. Maybe that is good. So that is what is called as maximum likelihood method. Likewise, you have this k-tuple method. So this k-tuple method, you have a window, right? So that is what is going to be the k-tuple method. Okay, so these are the different methods that are available for constructing your phylogenetic tree. Understood, everybody? So this maximum parsimony is only for similar sequences, and usually small number of sequences you take. And if it is going to be this maximum likelihood method, this is for dissimilar sequence or no idea sequence, right? So that only you go with the maximum likelihood method. And what about your NJ? It is distance based, no? So it is very faster. It is calculated. Whenever you are speaking with numerical, it is calculated. It is a fast method. Upjima, it is for related sequence. That means it is for similar sequence only it is going to work out. And Fitch method, it is less sensitive to variation. Okay. So these are the different methods that is available. Understood, everybody in the class? Any doubts, please do let me know on this. Clear? Shall we make up a move towards the next question? Yes or no? Come on everybody, cheer up, cheer up. Fantastic guys. Right. Yes. So the method of maximum parsimony is also known as, I think they directly extracted from full phylogenetics only. This is 2008, correct? So yes everyone. So this 2008 they have extracted direct from everything from your phylogenetics only. How many questions are coming? My God. Okay. So the method of maximum parsimony is also known as maximum evolution, minimum evolution, zero evolution, is it possible? Moderate evolution. Very good, Ansh. Yes, Kunal. Fantastic. Yes, Krishna. Good. Ram Prabha. Very good. Yes, Praveen. Fantastic. So, maximum parsimony means there is a least change. That is the key word I taught you. It is the least change. If it is going to be the least change, how should be the evolution? It should be minimum. Understood everybody? If at all I am saying that something is very least. Yes, so it is a very least change. So if it is going to be a least change, that means that evolution is not there. Minimum evolution. Understood and agreed? Right. Yes. Okay. So let's see few gate questions. So this might be as simple for you, but still no, we'll just have a look to have a revision. An example of derived protein structure database. An example of derived protein structure database is come on everybody. Derived protein structural database. Fantastic. Yes, Ash. Kunal. Structure. Okay. Good everyone. So first let us see how is all your database being classified. Okay. Alright. So majorly you have two major divisions of database. One, it is going to be the sequential database. Another one, it is going to be the structural database. Okay. Right. Under the sequence, you have two sequences. What are they? What are the two sequences? Nucleic acid sequence, protein sequence. Correct. But structure means only protein is there. Right? Yes. Fantastic. So under the nucleic acid sequence database, you have your NCBI, you have your DNA Yes, very good, Chiranjeevi. Yes, Deepankar. You have your DDBJ, you have your EMBL. So these are all coming under the nucleic acid database. Clear? Right. Under the protein, who is coming under the so what about PubMed? PubMed is nothing to do here. It is going to be the database. Yes, Deepankar. So this PubMed is only for literature. Yes, absolutely. Very good, Chiranjeevi. So it is NCBI, DDBJ, EMBL. Very good. Yes. So coming to the protein. Okay, so as far as the protein is considered, okay, so what will be the sequence database of protein? You have your, yes, very old friend, Swiss Prot. Okay, this has only now split into new names like Uniprot. Yes, absolutely, Uniprot. Very good. Yes, Deepankar. Yes, Ananta. So that is going to be Swiss Prot or Uniprot you call, and TREMBL. Yes, good. So already EMBL, European Molecular Biology Laboratory, it is there for nucleic acid. This tremble is nothing but translated EMBL. Understood? Fantastic. So this is all meant for your sequence, right? So they are all meant for your sequence. So this is going to be the primary sequence database. Okay, right. You have something called as secondary sequence database. Do you know what are they? Secondary sequence database. It is going to include the prosite. Okay, so the alignment or you are going to do some kind of categorization using sequence alone. You are not going to touch the structure. Only sequence alone you are going to make up. It is prosite, PFAM, very good. Then you have this blocks. Okay, so you have this identity. Yes, very good. You have this print, block, identity, PFAM, prosite, prodom. Okay, so these are all coming under the secondary sequence database. Secondary means it is all derived. The derived from, or you call it as metadata. Okay, yes, coming Kunal. PDB is structural. They do not come under the sequence. Okay. Clear? Right. This is all only the sequence. That means the primary structure. Okay, so they don't have, these are all the sequence. Right, so they don't contain any structure. It is all going to be the sequence. Yes, right. So in case of the secondary one, see, for example, if at all you are reading an original research article, yes, you are reading a review. Okay, right. So what do you mean by review? Will you write each and every, will you experiment each and every sentence? No, you are going to derive the information from the original article. Correct? You are going to simply compile the information from the original research. Isn't it? That is what honestly it is called as a review. Correct? So that is what it is happening in the secondary sequence database. I will not work directly, or if at all it is going to be the Swiss Prot or the Uniprot or TREMBL, the authors will directly submit the data. So what is the origin of all the sequence? Maybe you could have performed an N-terminal sequencing. Yes, maybe you could have done a Malditov MS, right? Maybe you could have done the Standam MS. Experimental data is what is fed into this primary sequence database. Okay, in secondary sequence, we do not do any wet lab. What you do is that you gather all the information from already existing database. Okay, so you will get an idea. Alright, so I am going to simply do a review. Okay, so for example, many people could have done research on COVID. I'm going to compile a review. So how the COVID was prevalent in India versus Europe. Okay, so I'm not going to do any wet lab, but I'm going to simply review the data. This is what is called as derived data. Understood everybody in the class? Yes, Chiranjeevi, now you got it. What is a derived data? Yes? Okay. So the same thing is applied for this derived secondary sequence. That means you are not going to prepare any secondary sequence. Absolutely. Correct. So whenever you are going to derive something, whenever you are going to extract something, that becomes your secondary sequence database. Understood? Yes? So your ProSite, PFAM and all, it is the collection of information that you could have got from the Swiss Prot. Okay? So is this clear to you? Right? Okay. So now coming to the structural database. Under the structural database, under the protein you have the primary structural database. This is only going to be your PDB, protein data bank. So it will give you a 3D structure. Only one fellow is there. But the secondary structure database, or you call them as the secondary, or you call them as the derived structural database, it is going to include scope, cat. Okay, these two are the major players of derived structural database. What is the scope? It is going to be the structural classification of proteins. What is cat? Class, archaeology, topology, homology. Okay, so these two are going to be the derived structural database. Everybody in the class, did you now get a total idea? Take a snip, I am going to change the slide. Everybody in the class, is it clear to you? What is a sequence database? What is the structural database? What is the primary structure? What is the secondary structure? What is the primary sequence? What is the secondary sequence? Fantastic. Yes, everybody. So now you are going to tell me, what is PFAM? Very good. What is PFAM? PFAM is, thank you Krishna. PFAM is secondary sequence. One more time you want? Sure, Preeti. In a moment I will do. Secondary sequence. Okay. Right. What about ProSite? Same. It is also secondary sequence. Okay. So now what I will do, I will just ask the question and fill the slide. Okay. Right. So primary sequence database of protein. So it is going to include two. What are they? Fast, we are going to fill the table. Yes, quick. Primary sequence database of protein. It is going to be TREMBL as well as your UniProt or Swiss Prot, whatever you call by your name. That is fantastic. It is the primary sequence database. So what are the secondary sequence database of protein? It is going to include your PFAM. UniProt and PFAM is different, isn't it? So this is PFAM. This is going to be ProSite. This is going to be ProDom. Okay. It is going to be your block. It is going to be your identity. Yes. Print. Yes. Very good everybody. Keep it up. Too good. So these are the secondary sequence database of protein. Fantastic. Now what is going to be the primary structural database? Okay, primary means first hand information. You should not think primary means sequence, how this is coming as a primary structure. Primary means the original research. It is one and only PDB. Okay, and then who is the secondary structural database for protein? It is going to be your scope. Yes, secondary structural classification of protein. CAT, class, archaeology, topology, homology. Understood everybody? So now it is all clear? Yes? Okay, fantastic all. So this is all about your proteins. Many questions have come in the past regarding this. So yeah, so CAT is going to be the class, archaeology, topology and homology. From this you extract the information, you form this, it becomes the secondary. Clear? Right. So for this, this is only going to be the correct answer. Understood everybody? Okay. So see this question, it could be simple for you, 2019 question. Which one of the following is a database of protein sequence motif? Good clue, right? Protein sequence motif. Think and answer. I want the correct answer. Everybody in the class. Good. Yes, Akshi, Samantha. Come on, everybody. Think and answer. Fantastic. Yes. Everybody got it? Yes, Siranjeevi. Now you are going to tell me, what about PDB? Good, Kunal. What about PDB? PDB, it is a structural database. Yes, Ansh, very good. What about TREMBL? TREMBL, it is a sequence, isn't it? Swiss prot, this is also sequence and primary, isn't it? Among the sequence, it is primary. This is only secondary because you got a term called motif. All this motif, fold, domain. Okay, so all these are characteristics of our secondary. Understood everyone? So now did you get the point of this classification? Yes? Okay, fantastic. That's all, simple only. Okay. So see our DBT recent question. Recent means maybe I think this could be from 16 or 17 or 18. A representative sequence profile of a given nucleotide binding domain, okay, is used to be mine means search the related sequences from TREMBL, the database that is used to extract the query corresponding to this fold is which of the following? Interpro, PFAM, TREMBL, gene ontology. You want to find the thing. Yes, Kunal. Yes, Sunanta. Correct only. See, TREMBL is going to be your nucleotide database. Okay. So, to this nucleotide, who is binding? Profile. Profile means, it is going to be none other than your fold. Fold means it should be from your PFAM. Got the point? Everybody in the class, how it became B? So PFAM, it is where you find all the proteins. Okay. So this is very important fellow. He has all the information. Okay. Right. What is this interpro? They gave something called interpro. No. This interpro is nothing but it is providing your functional analysis of proteins. How they provide? By dividing them into families and predict their domains and their sites. Okay, so that is what is going to be this interpro. This interpro fellow, he is only called as signature. What is this interpro called as? Signature. Okay, so it is a very important fellow. Understood? Yes, everybody. So interpro is now clear. The answer is PFAM. You know what is Tremble and gene ontology, but interpro was new, no? So just why I just thought of putting you what is an interpro. Gene ontology, it is a resource tool. Okay, so for next question, the gene set enrichment analysis. Gene set enrichment analysis. Differentially expressed genes, it is grouped into broader functions. A typical tool or resource for this purpose is what? Gene ontology or BLAST against the nucleotide database, PFAM database, PRODOM database. So this is for genes. Okay. So which things you will eliminate first? Which one you will eliminate? Very good. Correct answer is going to be A. Yes, Kunal, Siranjeev, Vinash. Correct. The correct answer is A. But since because it is gene, PRODOM and PFORM or protein you will eliminate, isn't it? Right. And this is also no way related. The correct answer should be yes, Siranjeev. Correct. So the correct answer should be only A, isn't it? We know that PRODOM and all, it is for protein. Correct? Okay. So the next question, this is also 2008 DBT question. I can see that in 2008 bioinformatics have come as 12 questions. Okay, so it is like a very big contributor at that time. So anytime the trend may repeat, so you all please make intense care for preparing this. Just some 10 hours. Totally you just give 10 hours, this course will be completed. Okay, alright, simple course only. In a phylip is for phylogenetic tree. The first two line numbers indicate what? Actually, there is something called as the flat file. We could have studied this flat file for NCBI. How many of you remember this? We did this locus, gene identifier, version, CDs, yes or no? We did this or not? Likewise, for every experiment or every tool we use, we have this flat file. They are asking, yes Sanarnya, yes Kanal, in this phylip output, the first line, it is going to indicate what? In phylip, you don't worry about gaps. Okay, so you eliminate these two gaps. Come on. Yes. Yeah. So first it is going to be the number and then it is going to tell the length. So this is, yes Nikhil. So this is directly the flat file. Okay. Yeah, that's okay. That's what. So what you do, you have to understand the flat file for few things. Oh Nikhil, welcome to the session. So you have to just look at the flat file of NCBI. You have to look at the flat file of phylip and also your multiple sequence alignment. All the flat files you just have a look and go. That will be, you know, a better thing. Okay. Yes. That's okay. Fine. Yes. So the next one is, yeah, homology modeling. I told you that if it is going to be a homology modeling, it should have greater than 30% overall similarity. Understood? Yes. So it is going to have a 30% of overall similarity it should have. Right. So this homology modeling can be distinguished from ab initio. Alright. So before going into that point, I'll just tell you what are these. There are three major methods for modeling. Okay, right. So when you will model, yes Nikhil? Yes. Yes, Sunanta, translate and tell for me. Okay, right. So actually there are three methods for modeling. Okay, right. So there is going to be the 3D structure. Okay. Okay, so there is going to be the 3D structure, right? So you are going to predict the 3D structure. How you will predict? Number one, naturally you can diagnose it by using your X-ray. Yeah, so if X-ray crystallography is there, or if it is a very small fellow, NMR may help. Then you will get your X, Y, Z coordinate. Correct? So X, Y, Z coordinate angle will be there. Based on that, oh, is it? Okay. Yeah, you can write it down. Right. So this is going to be the X, Y, Z coordinate. By that, no, you have your pi angle, psi angle. No. Based on that, you can come up with the structure. Okay, so that is what you do in you view in your RASMol, PyMol. Okay, so these tools you can view it. Okay, by using these methods. Right, fantastic. But now you don't have any structure. Okay, so no structure is available. At this point, now you have to predict the structure. How you are going to predict it? You are going to use few methods. Okay, so among the methods, yes, 3 methods are very famous. Number 1, it is going to be homology modeling. That means, yes, you have a sequence, you have another sequence who has a structure. So wherever it is there, you can generally apply. Isn't it? Okay, so this fellow will also look like that only. Like that you do a comparative modeling. Homology means comparison. Okay, so he is identical. So you compare them and do a model. This you can do if 30% it is going to be homologous or similar. Okay, great. Actually not 30%. It should be 75% homologous. Then you can exactly copy the coordinate angles. There's the XYZ coordinate angles are there, no? That yeah, between your query and the target, 75% must be similar or identical. Then you copy the coordinate angles. Done. If at all it is little bit less, not 75, only 50% is there. Then you do something called threading. What do you mean by threading? See, this is your structure. Okay. Alright. So this sequence and this sequence are exactly matching. This one is different. So from this you can copy, maybe this region will look like this only. This region will look like this only. This you have no idea. So this alone you will use some other method to fill the gap. This is what is called as threading. Third one, if there is no similarity or very less than 35% similarity between your query and target, you are going to do this ab initio modeling. Ab initio modeling means for everything you are only going to generate the structure. Okay, so if this is going to be the sequence, maybe it is an alpha helix. If this is going, like we will be telling, no. So if it is going to be a helix maker, alpha helix breaker, correct? So if at all I am having prolin, can I put it into the alpha helix? No. Prolin is a helix breaker. So if prolin is present, what is the probability? Under which structure you can put it? From which structure you can put it? Prolin. It should be in the beta turns. For turns you require prolin. Yes, glycine and prolin, they must be present in the beta turn. Only 4 structure. 1, 2, 3, 4. Right? So in beta turn you require, like that. Individually you are going to think and put. If malic is present, if malic is present or maric is present, then you may put them as an alpha helix. Like that, individually you are going to think and put. So that is what is called as ab initio modeling. So what the difference between what is an ab initio modeling, what is a threading, what is a homology modeling? Everybody in the class? Good. Now, this homology modeling may be distinguished from that of the ab initio model. Because, why? How come? Yes, how come they are going to distinguish? Homology model requires a model to be built. Homology modeling requires alignment of target to the sample or a template. Homology modeling is usually applied to any protein sequence. The accuracy of model is independent of the percentage identity between the target and the template. Yes. So which could be correct? Homology modeling as well as think. How do you distinguish homology modeling from that of the ab initio? Good everyone, come on. Yes, absolutely, no doubt. B is going to be the correct answer. I told you, no. So in homology modeling, yes, Nanda, right? B only. In homology modeling, you are going to align. So I told you 30% overall similarity, and if you are going to align 75%, it should be matching. Correct? Yes. So it requires the alignment of the target to the template. So that is what is absolutely going to be the correct answer. And next one, final question maybe for the day. The molecular dynamic stimulation is carried out for, see, I think we started with 200 questions, now 210, almost 10 to 12 questions in 2008 paper alone. Okay, so molecular dynamic stimulation, it is carried out for obtaining the ensemble of structures at physiological condition, obtaining the structure at globular energy minimum, fitting prospective drug molecule to the receptor, modeling a protein sequence from the sequence alone. Very good. So stimulation, isn't it? Very good, Kunal. So stimulation means you want to make them at your physiological condition, isn't it? You stimulate something, you resemble, you make them at physiological condition how they will behave. Earlier, in the absence of your MD simulation, they were modeling something, but when it went inside the actual condition, it behaved differently. But they want to overcome the bridge. So that is why they make the proteins to behave in their physiological condition such that they can derive something out of it. Yes? Alright. So with this we will break today. So again in one more session we will be meeting back with more information. Okay. So please do follow me at Unacademy. Alright. So I will be conducting a number of good special classes for you. So these special classes are for free. Okay, so you can unlock them by using my code Sanarnya live. Okay, right. And then I do have this plus sessions. So this plus sessions are highly structured. You will be reaching the syllabus completion. Yes, thank you. So you will be reaching the syllabus completion. You can practice. Sure, Kunal, you can ping me. Yes, so you can practice the MCQs. Right, as well as you can revise with me, and absolutely you can take maximum mock tests with me, right? Okay, so please come up with a subscription and make the maximum use out of it. All the very best. Keep reading, keep revising. I'll catch you up in another interesting session. Block my time slot 2 to 3 p.m. Bye bye everybody. Take